Hello, this is Jordan.
And this is Quinn.
I want you to picture a completely sterile operating room for a second.
Okay.
You've got stainless steel surfaces, surgeons scrubbed from head to toe, millions of dollars of advanced medical technology monitoring every vital sign.
Right. Highly controlled.
Exactly. An environment that is totally isolated from the chaos of the natural world. Now,
Hmm.
take that image and contrast it with, well, a wooden box sitting out in a muddy field in New Zealand, just swarming with thousands of stinging insects.
It is quite the contrast.
It really is. Today, our exploration is entirely bridging the gap between that rugged, large-scale commercial agriculture and those pristine, highly engineered hospital environments.
Yeah.
We are looking at how a very specific type of honey goes from being just a raw agricultural product out in the dirt to becoming a highly regulated medical-grade wound care technology. And before we get too far into the weeds, I want to make sure you know that you can find more info about what we are talking about at Manuka and Wound Science by visiting manukawoundscience.org.
It really represents a massive paradigm shift in modern medicine.
Oh, for sure.
You are taking something fundamentally biological. I mean, something manufactured by insects, foraging in the wild, and applying it to the most critical, severe trauma care we have.
And that contrast is just wild to me. Look, for you listening, I actually run a commercial apiary operation out in North Dakota.
Which is not exactly a sterile environment.
Not at all. My daily world is anything but sterile. I'm managing thousands of colonies, sweating in a protective suit, dealing with wild weather variables.
Right.
I am constantly mixing up massive batches of what we call pollen sub, which is essentially artificial protein patties we feed the hives to keep them strong through the harsh seasons.
Yeah, it's very hands-on.
It's rough work, it's sticky, it's messy, and it is largely at the mercy of nature. So the idea of taking a product from a wooden box full of stinging insects and applying it directly onto an open surgical wound.
It sounds counterintuitive.
Exactly. It requires a level of hyper-precise clinical standard that is honestly staggering to a commercial producer like me.
Well, the journey from that muddy hive to the pristine hospital bed is fascinating. But it actually has to start before the honey is even made.
Wait, before it's made?
Yeah, it starts with the exact dirt those bees are flying over, because before we even look at the clinical research or the medical applications, we have to understand the terroir.
Like with wine.
Exactly like a fine wine. The geography dictates the medicine here.
Right, so you're saying nature does the initial heavy lifting. If the dirt matters so much, what kind of specific environments are we actually looking at?
So the data focuses heavily on the North Island of New Zealand. There is a research consortium there that monitors 12 highly protected apiaries through what they call the bee sanctuary program.
Okay.
And they aren't just out there checking on the health of the bees. They are rigorously tracking the microenvironmental factors.
Like?
They monitor the soil pH, the specific mineral content in the dirt, and the flowering density of the Manuka bush, which is scientifically known as Leptospermum scoparium.
And what kind of differences are they actually seeing in the honey based on those, dirt metrics?
The data is staggering, honestly. They averaged harvests across 47 monitored hives to look at the precursor to a compound called methylglycol, or MGO.
MGO, right?
Yeah, MGO is the primary active compound that gives this specific honey its incredible medical potency.
Okay, so what did the numbers look like?
Well, if you look at the coastal regions, specifically the Coromandel Peninsula, you have these maritime micronutrients blowing in from the ocean spray.
Sure.
And a very specific, slightly acidic soil pH of 5.8. Hives sitting in that specific coastal spot produce a massive 785 milligrams per kilogram of MGO.
Wow, 785. That is a huge concentration. But what happens if you move those bees away from the ocean?
If you just move slightly inland to the volcanic soils of the Rotorua region, where the soil pH bumps up slightly to 6.1, the MGO concentration drops.
Drops to what?
Down to 620 milligrams per kilogram.
Oh, wow.
Yeah, and if you move again further into the agricultural hill country areas in the Waikato region, where the pH goes up to 6.3, the potency drops even further down to 440 milligrams per kilogram.
That's a huge drop-off. And to even monitor that, our exploration today mentions they are actually testing the raw nectar samples using high-performance liquid chromatography.
Right.
and doing it within 48 hours of extraction just to establish that baseline MGO.
Which is a highly complex process.
Mm.
High-performance liquid chromatography, or HPLC,
Yeah.
essentially means they are forcing the liquid nectar through a highly pressurized tube,
Okay.
and it separates, identifies, and counts every single microscopic molecule.
Hmm.
Tells them exactly how potent the nectar is almost the moment the bee drops it off.
I mean, from a beekeeping perspective, trying to achieve that level of floral consistency is mind-blowing.
How so?
Well, it reminds me of the strict geographic isolation required for queen breeding, or, the constant battle we fight to prevent bee drifting.
Oh, how does drifting impact the consistency of a hive?
Well, if you've never been around a commercial yard, drifting is when a foraging bee leaves the hive to find nectar, gets blown off course by the wind, or just gets confused in a crowded apiary,
Right.
and then she returns to the wrong box. In a commercial yard with hundreds of hives stacked together, it happens all the time.
I can imagine.
It is incredibly difficult to control exactly where a forager goes and what she brings home. You can place a hive right next to a pristine field of blooming clover, but a bee might fly 3 miles in the opposite direction.
Just because she found something else?
Yeah, because she found a patch of weeds she thinks smells sweeter. So to have these protected New Zealand sanctuaries where you are guaranteeing the floral purity of the Manuka bush, I mean, it's a monumental logistical effort.
And that logistical effort is entirely necessary because the biology of those coastal maritime environments is pushing that MgO concentration 30 to 40% higher than the inland sites. Those maritime micronutrients in the soil directly alter the plant's metabolism, which changes the chemical makeup of the nectar the bees collect.
That makes sense.
And here's the clinical threshold. Only batches of honey that exceed 400 milligrams per kilogram of MgO pre-sterilization are even considered for medical processing.
Wow. So everything below that line is just rejected for clinical use.
Entirely rejected.
All right, so let's say you've harvested this incredibly potent, perfect honey from the coast. It tests well above that 400 MgO threshold.
Okay.
My instinct as a beekeeper is that nature has already done the hard work. Why can't a doctor just open the jar and apply it directly to a severe burn or a diabetic ulcer?
Because of the inherent hidden dangers of raw agricultural products.
Like what?
Specifically, the presence of Clostridium botulinum spores.
Oh, botulism.
Exactly. In an adult human's digestive tract, ingesting a few botulism spores in your morning tea with raw honey is generally completely harmless.
Right, your stomach acid destroys them.
Exactly. But if you take those same microscopic spores and introduce them directly into an open traumatic wound, into compromised tissue that lacks oxygen, you are creating the ultimate incubation chamber for a life-threatening, flesh-destroying infection.
So it absolutely has to be sterilized.
It has to be.
And the data details that the sterilization process relies on gamma irradiation. It requires 25 to 50 kilograms of exposure.
Which is significant.
Yeah. This neutralizes the bacterial spores and achieves international ISO 1111-137 standards for completely wiping out living microbes. But I have to push back hard on this.
Yeah, why?
Wait. If you blast raw honey with up to 50 kilograms of radiation to kill botulism spores, wouldn't that absolutely destroy the MgO and all the delicate enzymes that actually do the healing?
That's a common thought, yeah.
Because as a commercial producer, it is beaten to our heads to never even heat our honey extraction tanks above 100 degrees Fahrenheit.
Because of the enzymes.
Right. Heat instantly degrades those beneficial enzymes. Blasting it with radiation sounds like total overkill that would just ruin the medicine.
It's completely natural to assume that. But here is where the physics of the sterilization are actually brilliant. Gamma irradiation is essentially cold sterilization.
Cold?
Yeah, it doesn't generate heat like a commercial pasteurization or boiling process would.
So it's not cooking the honey?
Not at all. Gamma rays are high-energy photons. When they pass through the thick honey, think of bacterial DNA, like the botulism spores, as a massive, fragile, highly tangled ball of yarn.
Okay, a ball of yarn.
And think of the MgO molecules as tiny, solid glass marbles.
Got it.
When you fire high-energy radiation through the gel, the massive ball of yarn catches all that destructive energy and completely shatters.
Wow.
The DNA is destroyed, meaning the microbe can never replicate or harm the patient.
But what about the MgO marble?
Well, the tiny glass marble is so small and its molecular structure is so incredibly stable that the energy just passes right by it. It doesn't shatter.
Wait, really? Yeah.
Post-irradiation testing confirms that the MgO stability only sees a tiny variance of plus or minus 5%.
That's practically nothing.
Exactly. So medical engineers achieve total sterility, making it incredibly safe to pack into a deep surgical wound without destroying the very chemical mechanism that makes the honey medically valuable in the first place.
That clears up so much. Okay, so now we have a biologically active, wildly potent, but perfectly sterile medical gel.
Right.
Let's look at the actual physical mechanism taking place inside the wound. How does putting a sticky, sterile syrup onto damaged tissue actually heal it?
It all comes down to creating an osmotic vacuum.
An osmotic vacuum.
Yes. Medical honey has a very specific low water activity. Clinically, it's measured between zero point 0.5 and zero point 0.6 Ah,
Okay.
and it has an incredibly high sugar density, sitting at about 80%. To put it simply, at a molecular level, that heavy concentration of sugar is incredibly thirsty.
She's thirsty for water.
Exactly. It is desperate to bond with water.
You know, I can actually relate perfectly to that zero point 0.5 to zero point 0.6 water activity measurement.
Oh, really?
Yeah, in our commercial extraction rooms, water activity is everything. If the honey we pull from the hives is too wet, the ambient yeasts in the air will cause it to ferment,
Right, which ruins it.
bubbles up, and ruins the whole crop. But if it is too dry, it gets so incredibly thick and viscous that it won't even flow out of the extractor or push through the hoses.
That makes total sense.
It seems like the wound environment requires that exact same delicate balance.
That is the perfect way to look at it. Inside a wound, if there is too much moisture in the medical gel, it dilutes that osmotic gradient.
It quenches the sugar's thirst.
Exactly, and ruins the vacuum effect. But if it is too thick, it stops the fluid exchange altogether.
So how does that thirsty sugar vacuum actually work on the damaged human tissue?
Well, because the honey is so starved for water, nature wants to find an equilibrium. When it's packed into a wound, the honey acts like a microscopic sponge.
Okay.
It physically draws lymphatic fluid upward from the deeper tissue layers via passive diffusion. And this continuous upward flow of fluid naturally floats necrotic, dead tissue debris and hidden bacteria right up to the surface.
That's amazing.
It's a process called autolytic debridement. Instead of a surgeon having to take a scalpel and mechanically scrape away the dead tissue,
Which sounds awful.
it is agonizingly painful and damages the surrounding healthy cells. So instead of that, the fluid vacuum just gently lifts the bad stuff away.
That is an incredible visual. And while it is vacuuming out the bad stuff, what is it doing to the healthy tissue left behind?
This is where we see something called the Bohr effect.
The Bohr effect.
Yeah, the medical honey has a highly acidic pH sitting between 3.2 and 4.5. When that acidic environment is introduced to the human tissue, it triggers a reaction in the blood.
Okay, what kind of reaction?
Think of your red blood cells like little delivery trucks carrying oxygen.
Sure.
When they hit a highly acidic zone, the acid forces the hemoglobin to lose its grip and dump its cargo. So you are actively feeding the surrounding damaged tissue with a massive rush of localized oxygen, which is absolutely critical for rapid cellular repair.
And the healing doesn't stop there because the data outlines how the MGO acts on the bacteria that the vacuum just pulled up to the surface.
Right, the protein glycation.
Yeah, it mentioned this process called protein glycation. The MGO actively destroys biofilm-forming organisms like Staphylococcus aureus and Pseudomonas aeruginosa.
We really have to talk about biofilms because they are the absolute nightmare of modern hospitals.
I've heard they're tough to beat.
They are. Biofilms are essentially a slimy, impenetrable fortress of armor that bacteria build around themselves to hide from antibiotics.
Wow.
Staph aureus and Pseudomonas are notorious for building these fortresses in chronic, non-healing wounds. Standard antibiotics just bounce right off the slime layer.
But the MGO breaks through it.
Aggressively, MGO attacks these organisms through that protein glycation.
How does that work?
This essentially means the MGO molecules bind directly to the structural proteins of the bacteria's fortress and deform them.
So it breaks the walls down.
It completely gums up the works, tearing apart their protective biofilms and leaving the pathogens exposed and destroyed.
So because this entire mechanism relies so heavily on MGO destroying those bacterial fortresses, the clinical research shows that doctors actually have to prescribe different strengths of honey.
Right.
Depending on the severity of the trauma, you can't just use a one-size-fits-all tube of gel.
No, not at all. Clinicians use a highly specific MGO evidence matrix to match the potency to the trauma.
Let's break down that matrix. At the baseline clinical level, you have MGO 400 to 550.
Exactly.
This tier is used for superficial minor burns, first and second degree, and clean surgical sites.
And the application frequency is usually every 24 to 48 hours with a dressing change. It controls the baseline bacterial load and keeps inflammation down.
Then, as the physical trauma increases, the doctor steps up the potency.
Right. MgO 550 to 700 is utilized for partial thickness wounds, stage 2 and stage 3 pressure ulcers, and venous leg ulcers.
Okay, so deeper wounds.
Yeah, this is where the patient really needs that autolytic debridement we just talked about, actively pulling out the dead tissue and promoting the growth of new connective tissue.
Got it.
For these more severe cases, application is often required every 12 to 24 hours, depending on how much fluid the wound is weeping.
And then you get to the heavy artillery, MgO 850 and above.
Yes.
This level is prescribed for deep tissue wounds, severe diabetic ulcers, and wounds positive for MRSA.
The superbugs.
Exactly. This tier provides aggressive, heavy biofilm disruption. You are dealing with multi-drug resistant superbugs, and the absolute highest MgO concentration is required to break through those stubborn bacterial defenses.
And if we look beyond just topical wound gels, the cutting-edge clinical research coming out right now is taking this MgO matrix into entirely new medical frontiers.
The recent studies mentioned in our exploration today blew my mind. They are literally using this clinical-grade honey on horse eyes,
Yes.
specifically treating a condition called equine ulcerative keratitis.
Treating severe bacterial infections directly on the delicate cornea of an animal, it's incredible. But the study that really stands out to me is from February 2026.
Oh, which one was that?
The one looking at synthesizing an antimicrobial honey-based composite bone cement for orthopedics.
Bone cement, like for joint replacements.
Yes. Think about why joint replacements or orthopedic surgeries fail.
Infection.
Exactly. They carry a massive risk of deep tissue biofilm infections because bacteria love to cling to the artificial metal hardware.
Oh, that makes sense.
By integrating the antimicrobial properties of MgO directly into the literal structural glue, the bone cement, they are preventing those biofilms from ever taking hold on the hardware in the first place.
That is brilliant.
It is a perfect collision of natural biology, apiology, and modern surgical physics. Researchers are also creating artificial skin-like films made of specialized polymers and gelatin and infusing them with these potent honeys to create functional antibacterial barriers for burn victims.
That is just next-level science. But right here, we absolutely must highlight a critical warning from the data.
Oh, absolutely.
We've talked a lot about MgO numbers, 400, 850. The reality is you can walk into a local grocery store right now and buy consumer-grade honeys.
Brands like Weatherspoon or Manuka Health.
Right. And they proudly display ratings from MgO 50+ all the way up to MgO 1100+ right on the label.
This is a vital, life-saving distinction to make for anyone listening.
Those consumer products in the grocery store are entirely unsterilized.
Completely.
They have not been through the gamma irradiation process. They are classified purely for dietary wellness, oral health, or as a preventive skin barrier on unbroken skin only.
Even if a jar of consumer honey says it has an MgO of 1100, which is the absolute highest potency available on the shelf, it still contains live microbial material.
Right.
It still contains the dormant botulism spores.
It must never be used in place of clinical-grade gel on an open wound, a surgical site, or a clinical ulcer.
Never.
If you try to use Pantry honey to treat a diabetic leg ulcer at home, you could introduce botulism into the wound and cause a catastrophic amputation or worse.
It's just not worth the risk.
Not at all. Clinical wound management requires sterile, irradiated products sourced specifically from specialized hospital supply vendors. Do not DIY your severe wound care with raw agricultural products.
Knowledge is only valuable when applied safely. And understanding that distinction brings us back to just how remarkable this entire highly engineered process really is.
It really is.
It actually brings up a concept that I think is worth sitting with. If something as subtle as the maritime air blowing off the ocean and a 0.3 shift in the soil pH of a coastal New Zealand peninsula, if that can fundamentally dictate whether a medical treatment has the potency to conquer a drug-resistant MRSA infection in a hospital across the world, it makes you wonder what else is out there.
What do you mean by that?
Well, think about the sheer scale of the natural world. If bees flying over slightly acidic, salty dirt can create a cure for hospital superbugs, what kind of undiscovered chemical combinations are wasps or ants currently mixing up right now in the Amazon rainforest?
Oh, wow. I never thought of it like that.
Right. We might just be scratching the absolute surface of insect-engineered medicine. How many of those future medical breakthroughs are entirely dependent on us maintaining hyperlocal environmental stewardship?
It really makes you look at a patch of dirt and, a muddy wooden box of bees in a completely different light.
It certainly does.
The margin between a failed agricultural crop and a revolutionary, life-saving medical device is razor thin, and it all starts with the soil. Remember, you can find more info about this at Manuka and Wound Science by visiting manukawoundscience.org.